Good afternoon. I'm Andrew Blake, the Vice President of Clare Hall. I'm very pleased to welcome you all, Clare Hall members, others from around the university, and friends from beyond the university. Thanks also to Robinson College for again letting us use this beautiful auditorium. This is the 6th King Lecture from Clare Hall, to be given by Professor Sir Shankar Balasubramanian of the Cambridge University. The King Lectures are a series in biomedical science, the third and most recent of our named series of lectures from the college, the first being the Tanner on Science and Human Values, which was established in 1978, followed by the Ashby on the Philosophy of Life and Society, established in 1985. And the current series, the King, was the generous gift of the pathologist, the late Professor Donald West King, who was an honorary fellow of Clare Hall and former executive vice president of the University of Chicago Medical Center. The founding lecturer in this series in 2016 was Harold Varmus, who got the Nobel Prize for the discovery of the origin of retroviral oncogenes. The most recent was in the end of 2023 from Jennifer Doudna, who got the Nobel Prize for the CRISPR-Cas9 gene editing technology, and several other very distinguished lecturers in between. The series was initiated by our own Dr. Laurie Passmore, who is a fellow of the college and head of division at the MRC Laboratory for Molecular Biology. And this year she's been joined by Dr. Madeleine Lancaster, also at the Laboratory for Molecular Biology, organizing the lecture. For myself, I'm very excited to be here to watch the lecture, and I'd like to invite to the podium Laurie Passmore to introduce the lecture.
Thank you, Andrew, and a very warm welcome to all of you. We're absolutely thrilled to have with us today Sir Shankar Balasubramanian as our 6th King Lecturer at Clare Hall. Sir Shankar is the Herchel Smith Professor of Medicinal Chemistry here at the University of Cambridge. He's a senior group leader at Cancer Research UK's Cambridge Institute, and he's a fellow of Trinity College. He works on the chemistry, structure, and function of nucleic acids. And as I'll tell you more about that in a minute, but this work has really, he's been awarded some of the highest accolades in life sciences based on this work. And so these include the Royal Medal of the Royal Society, the Millennium Technology Prize, the Breakthrough Prize for Life Sciences, and the Gairdner Prize for Life Sciences, some of those jointly with Sir David Klenerman and Pascal Meyer. Sir Shankar was knighted in the Queen's New Year's Honours in 2017 for his services to science and medicine. He's a Fellow of the Royal Society and an international member of the National Academy of Sciences. Shankar was born in India, but spent almost his entire life in the UK. He studied natural sciences here at the University of Cambridge and did his PhD with Chris Abel here in Cambridge on enzyme chemistry. After a postdoc at Pennsylvania State University, he returned to Cambridge in 1994 to start his own group. Just a few years after that, Shankar and David Klenerman, his colleague here in Cambridge, came up with an innovative new way to sequence DNA based on fluorescently labeled nucleotides and imaging. So they ran with this idea and founded a company called Solexa, just 1 year later, to develop the technology. Solexa was acquired by Illumina in 2007, and the result, the next generation sequencing technology, is now responsible for almost all DNA and RNA sequencing. It has truly revolutionized biology and medicine. So what started out really as a basic science blue skies research project led to technology that was many orders of magnitude faster and cheaper than previous technology, and it's highly scalable. So we can now sequence whole genomes routinely, so for humans in an hour or so, but we can also sequence genomes of any living organism, really allowing us to understand so much more about the biology and the environment. But Shankar doesn't only work on sequencing technologies. Some of his work focuses on epigenetics and specific nucleic acid structures called G-quadruplexes. Now he's studying these and other aspects of nucleic acid biology by developing small molecules that either stabilize, map, or modulate these structures. And so this work has revealed new biology, but is also leading to new therapeutic avenues to treat disease. He'll be giving a scientific lecture on G-quadruplexes tomorrow at the MRC Laboratory of Molecular Biology, to which you're welcome to join us. So I hope you can begin to appreciate the amazing combination of technology development, as well as chemistry and biology understanding that characterizes the unique and broad-ranging influence of Sir Shankar's work. So with that, I'd like to welcome Sir Shankar Balasubramanian to the stage for the 6th King Lecture.
Thank you, Laurie and Madeline and the fellows of Clare Hall for the honor of giving this lecture today. So I'm going to tell you a story that involved some basic science that David Klenerman and I were doing here in the university more than 25 years ago, the beginning of our independent careers. And that led to, as you've heard, we founded a company called Solexa to develop the technology based on what we'd done in the university. And then that got taken on by an American company called Illumina afterwards. So we have an audience of people from different backgrounds here. So I'm going to start with some basics. I've spent my whole career studying various aspects of DNA. I'm a chemist, so I always view things in terms of their chemical structure and how the chemical structure leads to properties. And in this case, DNA is an information molecule, and I'll say that we are still discovering new aspects of DNA that convey information and function that contributes to biology and also disease-related biology. So that's still an ongoing plan, and we tend to think of 71 years ago when Watson and Crick came out with this structure that was the beginning and the end of what we understand about DNA. That was really the beginning. Now, in the center of a DNA double helix, there are 2 strands, one in green and one in yellow, shown here. And they are linked by interactions shown here. So the C base is linked to the G base through 3 what we call hydrogen bonding interactions. And the T base and the A base is linked through 2 hydrogen bonding interactions. Now, these 2 types of interactions are, in essence, the molecular basis for the genetic code. It's remarkable, the complexity of biology boils down to 2 types of interactions between these 2 base pairs. Now, this is DNA shown in linear fashion, 2 strands here. A clicks with T and C clicks with G and vice versa. Now, one of your genomes in one of your cells, 1 copy of your genome comprises 3.2 billion of these building blocks. And the order in which they are linked together is what we call the DNA sequence. And the order in which they're linked together basically heralds all the information that contributes to your genes. Now, genes are stretches of letters, typically a few thousand of these letters, that carry the code that ultimately designs proteins, which are the building blocks and machines of living systems. So determining the order in which these building blocks are linked together has been a major endeavor in science. And the first well-used method to do this was created by Fred Sanger in Cambridge in the late 70s. That's called Sanger sequencing. And you will have known of the Sanger Institute, where the Human Genome Project was sequenced in part. Now, why sequence DNA? And my focus is going to be on human genome sequencing and the motivations for determining your genomes. Now, all living systems carry their code in the form of DNA. There are some retroviruses, such as SARS-CoV-2 that you will all know about, that carry its genome in the form of RNA, close relative of DNA. So in order to really fully understand a biological system, you do need to know what the sequence of the genome is. Now, in the case of humans, and this was really a primary driver behind Human Genome Project and also the expansion of this project, is to understand the genetic basis for what makes us different, what predisposes humans to certain diseases, requires in part that you need to understand the sequence of the genome. So it's absolutely fundamental to do this. Now, turning the clock back, can't believe it's 30 years ago now, actually, 1995. This is something that we were actually not doing. This is David Klenerman here, my partner in crime with this project. And these were the two research co-workers that we had in our lab working on this at the time. Colin was in my lab, he's an organic chemist, and Mark is a physical chemist, rather like David Klenerman. And I should say the way he and I came together, we started at the same time in the Department of Chemistry quite near here. And I was doing an experiment where I needed a laser of a certain wavelength and a certain type. And I didn't have one, so I walked around the building trying to find someone who had such a laser. And one of my colleagues said, you need to go and find this guy, Klenerman. He's in the basement in a room with no windows, and he has lots of lasers and things. You should talk to him. So I went to talk to David, and we did the experiment that I wanted, and then we kept talking. And we dreamt up a project together, just literally over a cup of coffee. And at that time, I was interested in molecular machines, enzymes that synthesize new copies of DNA. So every time your cells divide, somehow the cell needs to duplicate the genome accurately so that each daughter cell has its own copy. And the machines that do this are called DNA polymerases. Now, I was studying how these machines work, and the way they work is they take one strand, one of the two strands of DNA, and they build a complementary strand by adding one building block at a time. And it knows which building block to put in what order, because it reads the template and it ensures that it preserves the Watson-Crick rules of matching G with C and T with A, and so forth. So we were interested in this process. Now, David is a laser spectroscopist. And in the mid-1990s, there was a relatively new observation technique capable of detecting single molecules of fluorophores at room temperature. Now, this was new. And I should say, throughout science, new measurement techniques, new observation techniques have driven inflections in what we can do and what we can learn. And this was such a time. So we thought, why don't we bring together this new observation technique and watch one of these machines synthesize DNA in real time and see what we could learn. And that was all there was to it. We had no expectations of what we would learn, but we thought by applying a new technique, we would learn something new. Now, we wrote a grant that was 2.5 pages of A4 long, and it was funded purely on the basis of what I've just said. Quite remarkable, really. I still have that grant, actually, and every now and then I read it and somewhat in disbelief about the naivety of what we propose. Now, there were many different ways that we organized our experiment, and I'm going to tell you one of them. First, we had to attach a fluorophore somewhere because that was the observable. Without that, you wouldn't see anything. And we had to attach it in a way that allowed us to learn something about the mechanics of what was going on. Now, one of the ways we did this was to take a glass surface, it was fused silica, and then we were able to take DNA that we'd synthesized in the lab with a long template and a shorter piece that we call a primer, and attach that to this surface so it doesn't go away. And then we would bind the DNA polymerase to this DNA. And what this does is it incorporates building blocks one at a time. And we attached fluorescent molecules to the building blocks so that we could actually watch them go in as this strand of DNA was being extended by this machine. Now, in order to do this, we built in David's lab an instrument. I know there are some physical scientists here. It was a total internal reflection microscope with 2-color excitation. And we use that microscope to make these observations. And then we digressed somewhere nearby. This is local, so actually many of you will know this place. It's behind the chemistry department on Panton Street. And engineers, and I know there are some engineers here, engineers and chemists go there still quite frequently. And I used to go there very frequently, actually, and until I got progressively promoted. And at this stage in my career, this is a recent photo, but back then, David and I were junior. We weren't even lecturers. We were research fellows. I was a Royal Society research fellow. And actually, I started out with no office. Whenever we recruit young faculty now and they're negotiating their package, I sometimes tell them what my package was, which was no office and no lab and no money. And actually, the advantage of not having an office, and then I was given a small office that I shared with another junior faculty member, and one of us had to literally climb over the other one to get in or out of this office. So if we wanted to chat, we would go somewhere else, and so we used to go here. And what I've learned over the time of my career is I discovered a new equation, and that is the amount of time I spend here is inversely proportional to the area of my office in square meters. And I don't go there very often anymore. But here, we used to go there and digress a little bit. And it was during one of those digressions that we saw a way to adapt what we were doing to actually read the sequence of the DNA, which actually wasn't what we were trying to do. And we do this as follows. This was the thought experiment. If we take the DNA, here's the template, here's the primer, here's the surface. And instead of just having one fluorophore, if we could color code the 4 different building blocks so that the G has its own color, the C has its own color, and similarly for A and for T, then if we could somehow do this chemically, and if we could persuade one of these machines to tolerate the unnatural aspects of these building blocks, we set up a competition. And because of the Watson-Crick base pairing rules, the polymerase will choose the correct one of the 4 building blocks. In this case, the next templating base is a T, so it'll choose the A and it will incorporate it and not the other 3. Now, if we could stop this incorporation after a single building block, we could image this surface, and at this particular location on the surface, we would read a red color and decode 1 building block. Now, if we then carry out a second cycle, but first we would somehow have to detach this color because we've retrieved the information, and unblock this end so that we can then do it again, you would then bring in the T with its color in the second cycle. And in this way, we would successively build this strand, and you would see a succession of color changes. And the order of the color changes would reveal the sequence of this template. Now, that in itself doesn't provide any advantage over the Sanger sequencing method of its day. It's just a different format with the DNA attached to a surface. But the advantage of immobilizing your sample to a surface is that you can parallelize massively. And as we've seen in the electronics and computer chip industry, it's been driven by leaps and bounds due to miniaturization of features and parallelization. And so what we envisaged is that you could perhaps take the DNA from a cell, fragment the DNA into manageable pieces, and immobilize these fragments onto a surface. And if we could do this in a way that gave each piece of DNA optically resolvable space around it, you could then sequence all of these using this approach in parallel at the same time. So if you had a million different sequences on your surface, you would sequence them all in parallel. Again, this was the thought experiment. So we paid a visit actually at that time to the Wellcome Trust Sanger Institute to meet 3 of the sort of founding lead scientists of the UK part of the Human Genome Project. It was an important visit because it helped us contextualize the potential value of possibilities. And we'd carried out this calculation. A human genome was known to be of the order of 3 billion of these letters organized. So we made an assumption about what we could build an array. We looked at the kinetics of the individual reaction steps. And we thought we could build a system that could sequence of the order of 1 billion bases in one experiment. Now, at that time, the state of the art was of the order of 100,000 per experiment. So this was 10,000 times sort of bigger or faster than the current state of the art. So we discussed this with our colleagues at the Sanger Institute, and also had the question, somewhat facetious question. We said, we were just 2 young chemists who hadn't really achieved anything scientifically at that stage. But we asked the question, the Human Genome Project is one representative reference of the human genome. In order to find the genetic basis of who we are and predisposition, you have to do this on a population scale at some point. How are you going to do that? And I think at that point in time, there certainly wasn't a clear idea of scaling the Human Genome Project to populations. So we discussed our idea with them, and you know, the vision of maybe one day it could sequence at population scale. And so they gave us the endorsements that we needed to actually go and do something about it. Now, little bit of chemistry, because it is important to what was done. This is the sort of the chemical mechanism of how these machines work. Here's a template, and this is the part that's going to be extended one building block at a time. Now, the active site of this enzyme, the key part of the active site is actually these 2 amino acids that bring in 2 magnesiums. The magnesium ions are the catalyst for this reaction. And this oxygen here on the part of the DNA that is growing is critical. It acts as what chemists call a nucleophile. Now, this in red is a building block, and the base is just the letter. This can be a G or a C or a T or an A. And I haven't drawn the template strand, but the template strand runs here. And if the templating base is a T, it will attract an A here. And the enzyme catalyzes this reaction where it forms a bond between this oxygen and this phosphorus atom of the incoming building block to build this linkage called a phosphodiester linkage. So for the process, we had to hijack this natural chemical mechanism to suit our means. And the way we did this was by adding a fair amount of unnatural chemical structure. I'm not going to go into the details of this, but quite simply, the oxygen that is vital for each step of the reaction, we add a group that chemists call a protecting group on here. And what a protecting group is it's something that can stop reactivity, but you can remove it whenever you choose to by controlled chemistry. And we used similar chemical functionality to attach a fluorophore through the non-Watson-Crick edge of each of the 4 bases. And we used a chemistry that was invented in the early 1900s by a German chemist called Hermann Staudinger. And this is a reaction that can be harnessed to work in water at moderate temperature. And it is beautifully specific chemistry that does not attack or destroy all the complex chemical structure of DNA. And what it does is it cleaves this bond here, and it cleaves this bond here when you so choose. So this was a way of attaching fluorophores to each of the 4 bases. And what I haven't shown is that we did some careful re-engineering of the machines, the polymerases, to tolerate these changes. And once it is incorporated and we retrieve the information, you can lose the fluorophore and lose the protecting group to do the next cycle. Now, we were doing all this by detecting single molecules of DNA. Now, during the development work, we started to see some interesting stochastic effects of single molecule observation. Now, these are very interesting, but when you're sequencing, you want accuracy and you don't really want these stochastic effects. And many of you will have known the great Sidney Brenner, who spent quite a bit of time around these parts. Now, Sidney took an interest in what we were doing. And one day I was summoned to his house in Ely and had a conversation in his kitchen, surrounded by kettles and pots and pans and things. And he persuaded me that we should amplify these single molecules on a surface in order to get lots of representative copies, as this would iron out these stochastic effects that could lead to errors. They'd be very difficult to distinguish from natural errors. And so that was a very sound piece of advice that we adopted. And he also had an idea for amplifying them that we figured wouldn't work on our system, and he was less happy that we didn't adopt his idea. But we adopted a method that was developed by Pascal Meyer, is a French biophysicist, and he and his team had a parallel effort. And one of the things that they developed was a method which I'll schematically show you here. And this is actually what is now part of the Illumina systems for amplifying the sample array. So we generate a single molecule array. Now, here's a fragment of DNA. You can use standard molecular biology and synthetic DNA to add known sequences to either end or both ends. Now, the surface we coat with synthetic short DNA fragments of a sequence that's complementary to one end or the other end or both ends, so that when you flow in these fragments, they bind by Watson-Crick base pairing to the surface. Now, the method that Pascal and his team developed, we call bridge amplification, and that is if you melt one of the strands, you can anneal the other end of your sample fragment to another primer on the surface. Now, if you add a polymerase and natural building blocks, you can make a copy strand. And in this way, you can go through cycles of melting and re-annealing and extension. And because one end of the DNA is always anchored on the surface, you end up with identical copies in the same space on the surface. And then you can apply solid phase sequencing, where each cycle you get fluorescence from lots of molecules. In fact, it's hundreds of copies at that site. So this was an important part of the invention that was an add-on. It was different from our original vision. Now, in 2005, in Solexa was the first small genome that was sequenced. A tiny genome, in fact, of bacteriophage 5X174. And this is a CCD camera image of the surface. This is an expansion, and each of these circles here is what we call a cluster of DNA sample molecules, where the fragment has been amplified, and we've incorporated one fluorescent building block. And you'll see that these circles are one of 4 colors, and the color tells us the identity of the base. Now, the chemistry that's applied next removes all the color and resets the system in the n+1 position. And in the next cycle, at each of these sites, you will see a color that's indicative of the next base. So cycle by cycle, you build up a read of DNA information, and you're doing it in a massively parallel fashion. Now, one of the challenges or belief gaps to be jumped early on was we weren't sure how long a piece of DNA we could read. But calculations told us that if we could read at least 25 bases, and we don't know where they came from, given the Human Genome Project was going to generate a reference sequence, we could computationally align these fragments, and even if there was 1 or 2 unknown point mutations, that we would still be able to align these fragments and find out where they came from. So what happens is these fragments are computationally searching the reference genome for a nearest match. And then when you find it, you align there. And you oversample, so you align many different sample fragments of DNA, and then you can read each base with a high degree of confidence and accuracy. Now, the way genomes are typically sequenced today using this approach is called paired end sequencing, where you bind a primer to one end and you read typically 250 bases in one direction. And then you start reading from the other end about 250 bases. And then you align this to the reference. So in the summer of 2006, Solexa launched this system. It was called a 1G genome analyzer. And at the push of a button, it would sequence 1 billion bases of DNA. Over a period of a few weeks, I should say, it took a bit of time to get through this, but that was a landmark, and it actually matched our projection. You remember the little calculation I told you, that was 1 billion bases. So this was a commercial system. And the first machines ended up in places like the Sanger Institute and the Broad Institute in Boston. Now, in January 2007, a company called Illumina acquired the technology and the company. And I stayed involved with Illumina till 2017. And they continued to engineer and optimize and improve the technology. And by 2021, they produced systems that looked a little bit like this. And there are the modern versions of this that have come out in 2024. Now, these systems are production human genome sequencing instruments, and they typically sequence of the order of several trillion bases. And one of the main dimensions was actually reducing the features even more and packing them more tightly. So the density of information on the chip has progressively improved. That's one of the main improvements. So Laurie mentioned genome an hour. That's actually what these systems do. They'll sequence a genome 30 to 50-fold depth, about 1 hour. And I think the latest instruments will do tens of thousands of human genomes per year. So this is about it's actually approaching a 10 million-fold improvement on where things were when we started. Okay, I'm going to come back to a little bit about the types of contributions and impact some of you in the audience have made using this technology, actually. Just want to sort of pause to give a little bit of a plug for my lecture tomorrow. So I do work on other aspects of DNA. Now, one of them is a quadruple helix structure called a G-quadruplex. And if you want to know about that, you have to come and hear a different lecture tomorrow. But I will say a little bit about this on the right here, epigenetic DNA letters, and there are some members of the audience who know far more about this than I do, but my interest was piqued by the realization that DNA has more than 4 letters, as it were. Now, the genetics provides the fabric for the design of the machines that make up living systems. It's actually more complicated than that, because most of the genome is DNA that doesn't do that. The non-coding genome, which when we found over the last sort of 15, 20 years, a non-coding genome is important for regulating the coding genome. But there are also dynamic aspects of DNA building blocks. For example, in humans, the letter C can acquire a methyl group here. This is 5-methyl-C. And in 2009, it was shown in human cells that this can be further modified to form this 5-hydroxymethyl C, which is almost like a 6th letter. Now, these two variations on C are relatively dynamic, and they change and help orchestrate the extent to which genes are active or inactive. And this is hugely important for defining different cell functions, where cells have, in essence, the same genetic sequence, or they have a different functionality because they have a different program running the activation of genes. And with these epigenetic letters, they manifest in what I consider the major groove code. So here's a double helix. And there are external features. There are two grooves, one minor and one larger one called the major groove. And actually, I started by talking about the Watson-Crick recognition through these hydrogen bonds. If you look at the DNA through the major groove, you also see hydrogen bond donation, acceptor, acceptor, donation, acceptor, donation, acceptor. You see patterns of hydrogen bonding that are very specific in the major groove. And in nature, there are proteins that operate DNA. These include proteins called transcription factors and chromatin remodelers. And they are actually the machines that activate genes or inactivate genes in a very, very well orchestrated way. And these epigenetic letters, these epigenetic changes can be inserted. For example, you can have a change that in essence is a red signal that can lead to inactivation of genes. Or it can change again to something that is relatively more active state of genes. And it does so at the molecular level by dynamically altering how proteins read the double helix. So this is a dynamic aspect, and I think epigenetics is a burgeoning field, and my own involvement there has been to help generate sequencing chemistries that read, well, now there's a 6-letter genome that can be read. So you can get genetics and epigenetics. So I'll say little more about epigenetics and jump straight to the real reason why David and I were motivated to try and pursue the technology some 28 years ago. And that is, I think a lot of people in genetics realize the potential for impact in healthcare and medicine through better understanding our genomes. I'm going to give a few examples to round off with. I'm going to start with a very simple example, therapy selection. Now, BRAF is a gene that encodes for a protein that's involved in signaling, a signaling cascade that can lead to uncontrolled proliferation of cells. And there is a very specific mutation where a greasy amino acid in this protein is converted to an acidic charged amino acid. And the effect of this mutation is that this protein signals without getting its natural molecular cue. And so it drives uncontrolled cell proliferation. And there are certain cancers, and particularly a subset of melanomas, skin cancers, where this precise mutation is the underlying cause. Now, for those cancers, the physician will prescribe a drug that acts on this target. So and those patients are very responsive to the drug. So this is an example of genomic medicine. You make a diagnosis, you look at a very specific genetic trait that tells you about the underlying mechanism driving the tumor, and you treat it very precisely. So that is personalized genomic medicine, and this is one of the early and more straightforward examples. Now, there are projects that have carried out sequencing at scale. This is one example in the cancer area. Now, Genomics England, some of you will know about. This was actually started by former Prime Minister David Cameron. He was one of the founders of this. Genomics England was a project through the NHS where initially 100,000 patients, mainly in the area of cancer, infectious diseases and rare diseases, were sequenced as part of their treatment protocol in order to help us learn about the importance of genetic variation in those disease areas. Finland has a population of 5 million people. 10% of that population have had their genome sequenced, and this is all linked to their healthcare records, and it's a continuous program of learning. Now in the US, this All of Us program is seeking to sequence 1 million people. So population scale genome sequencing is happening. And I think there are over 1 million human genome sequences that have been done to date. Another area, early detection, and this is particularly burgeoning in the cancer area. It actually started with non-invasive prenatal testing. So this is something that Dennis Lowe in Hong Kong kicked off by the discovery that DNA from the yet-to-be-born child passes the placental barrier into the plasma of the mother. And so you can genetically test without having to carry out an amniocentesis, which has its own risks. Now, later on, a number of labs found that tumors release their DNA into the plasma. And if you take a blood draw and you isolate cell-free DNA fragments, and you have to sequence at great depth, it is a bit of a needle in a haystack challenge. You can find fragments of DNA that show the hallmarks of the tumor just from a blood test. And interestingly, the most prognostic hallmarks are epigenetic features rather than genetic features. And so there are a number of clinical trials going on now, including one in the NHS, to understand to what extent this approach might be used for surveillance and potential early detection of cancers through a simple non-invasive test. Now, I'm going to give you a cancer example. This is a very early study that was published in 2010, and it was actually carried out at UBC in Vancouver. And I learned recently that the patient involved was actually a faculty member there who had cancer of the tongue, and the standard of care stopped working. It's a common problem in cancer, resistance acquisition. And the team there carried out a pioneering study, and they sequenced some normal representative cells from the patient and the primary tumor and the metastasized tumor. And they looked at the data to see if they could learn anything about the underlying mechanics. And in cancers, especially advanced cancers, you see quite a lot of rearrangements in the genome that affect how the machinery is expressed. And they looked at some cancer driver pathways, and you often see genes are amplified, they're multiplied. Where there was one gene, there are now more than one. And if these genes are active, you end up overdosing with that gene. So they saw that. In particular, this gene was amplified, and therefore it was overdosed, and this pathway was overactive compared to normal. Now you can also see the opposite. You can see genes deleted or mutated, so they are inactive. And in these cases, the function of these genes goes down. Now what they saw is that this pathway and this gene product that was being treated with therapy was no longer active. It explained why the drug was no longer working. But this pathway was now active because this gene had been amplified. And in the study, they then treated this gene product with a known drug. And they were able to control the tumor for a little while. And then the tumor evolved again, and they carried out a similar process and treated with a different drug. Now, they extended the life of this patient, but it certainly wasn't a cure. But in the study, what the authors concluded was if they'd known at the beginning what they knew at the end, they would have probably prescribed 3 drugs in combination in a very rational way. And Mike Stratton is in the audience, and he's one of the leaders who've carried out extensive whole genome analysis of many tumors. And what studies such as these have done is taught us about commonly emergent patterns of rearrangements that occur in certain tumors. And in many clinics now, these patterns are actually being used to guide the type of therapy that's prescribed, because the patterns inform us about the mechanisms that are predominating in that tumor type. So that's an ongoing project in the cancer area. Now, rare diseases is another area of major study, and there's been a lot in the UK in the Genomics England-based rare diseases project. Now, I should say rare diseases are called this because they are difficult to characterize, and they have unusual features that are not that common. Collectively, 1 in 17 people have a rare disease. So as a collective, they're pretty common. They are typically conditions that manifest in early childhood as developmental disorders. And they are, to a very large extent, genetic in origin. Now, again, it's an old story, and this is work from Stephen Kingsmore. And this is a case where 2 parents bring their child into a pediatric clinic, and the child has obvious developmental problems. And the parents have gone through a series of tests and ruled out a series of known conditions. So they know what the child does not have. And here they sequenced what's called a trio. They sequenced mum, dad, and child. And because of the child's genome is inherited from a combination of both parental genomes. So computationally, it's relatively easy from this trio to find differences that are unique to the child. So that's what they did. They took blood samples, sent them over to Illumina, Cambridge. They were all sequenced. The data was sent back to the US, and the clinicians looked at the data, and they were able to sort of triage systematically what the possibilities may be. and what the genes that are causing the condition may be. And they were able to hone in on this particular gene, which is responsible for copper transportation and copper metabolism. So they diagnosed this as a metabolic disease. Now, such metabolic diseases are actually treatable if you understand what they are. This took 4 days. And if you look in the Guinness Book of Records, this same team are in there because they've now done this in less than 24 hours to generate a diagnosis. Now, these aren't rare stories. In Genomics England, they have sequenced tens of thousands of cases of rare diseases. And I think the stats are something between 25% and 30% of these cases. They are able to make a diagnosis that is scientific and mechanistic and exact. And a portion of those cases they're able to treat. And I think the remainder they are not yet able to treat, but understanding the mechanistic basis is the start of finding a cure. So I think in the case of rare diseases, there's substantial impact. I'll end with infectious diseases. Pathogens have genomes, and understanding the genome of the pathogen tells us about the nature of what we're up against. In January 2020, in Wuhan, in China, a team sequenced and identified the SARS-CoV-2 virus. Now, that was something called metagenomics, where you take a sample. I think this was a lung aspirate. It's got lots of different microorganisms. You extract the DNA of all of them and sequence it all, and then you computationally piece together the genomes, and they identified SARS-CoV-2. Now, this was a paper published in Nature, I think it was around June of 2020. By this stage, the virus had spread and started mutating. And the first line of this says more than 1 million SARS-CoV-2 genome sequences have been shared. They were taking blood samples from patients who were affected, and through sequencing these samples, they could identify the virus and also variants. The virus keeps mutating. And so there was a global collaboration where this was being deposited in real time, and there was a real-time geogenetic map of the emergence and spread of the virus and variants of the virus that was shared, that I think was helpful for understanding control. And also this sequence data was helpful in re-optimizing the mRNA vaccines, of which there have been several generations now, by seeing how the virus is mutating. So that's infectious diseases. So what I would say is we're still at a relatively early phase of seeing how rapid, low-cost sequencing will have an impact on healthcare. And I think over the next decade, we will start to understand the full extent of what's possible. Now, there are so many people who contributed to all aspects of what I've shared. The project started in the chemistry department in Cambridge, and I have to thank all of our co-workers involved with that. Many brilliant scientists in Solexa and also Illumina, scientists, engineers, mathematicians, molecular biologists, chemists, all manner of talents. Just end with these comments. This all started out as basic science. We weren't trying to do anything useful back then. It was, of course, a big team thing. But lastly, I think there are many examples now of scientists working as a collective to help address major problems. Thank you very much.
Thank you very much, Shankar, for an excellent lecture. It was really inspiring to hear how you went from talking about chemistry in the pub to all the way to medical diagnosis. So I'm sure there's some questions from the audience. We have some microphone runners on the sides.
Yeah, I was wondering, your 1995 grant, do you think it's more or less likely that it would be funded these days?
It's a political question. The general feeling is that science directives are less likely to fund fundamental science than before. I'm not sure if it's true, actually, because remember, grant funding is decided by us. And certainly in situations when I've been on the other side, sitting around a table with colleagues, deciding what to support, I think there's still a very healthy appetite to support creative ideas that are aimed at discovery and knowledge building without necessarily judging them on whether they have some immediate utility. So I think a lot of the sort of more superficial rhetoric suggests not, but I don't think it's too terrible. I think we still fund a lot of fundamental science, actually, in the UK.
Thanks, Shankar. That was wonderful. So I first heard you give a talk about being able to massively sequence DNA. It's nearly 20 years ago, and I remember you made a prediction that you said that in our laboratories we would all have a machine, which meant that we could switch it on and we could go for lunch, and when we came back, we would have all the DNA sequence we wanted. And I didn't really believe you at the time. And I thought it was a gross exaggeration. And why would I ever want that machine? And you were absolutely right. And now I have one in my lab. So my question is, where are we going to be in 20 years' time in terms of both technology and how you think we're going to use that technology?
Thanks, Paul. I think methods for observation, imaging, measuring molecular features in living systems is going to continue to evolve and improve, actually. And impossible to tell where we'll be in 20 years' time, but the current trends I see are being able to simultaneously measure different molecular features at single cell or single molecule or single fragment level. And we're doing some of this in my own lab, where we can measure genetics, DNA epigenetics, aspects of chromatin features, and protein interactions from the same fragment at the same time on a sort of whole genome or whole cell scale. So I think those sorts of things will become more accessible. It's still a little bit specialized. I think characterizing systems not in 2D, but in 3D molecularly will be realized over 10 to 20 years on a way that's practical. I think the same is true for imaging. I think sort of cryotomography is evolving, and I think getting close to atomic resolution imaging of living systems in 3D with all complexity is potential vision for 20 years' time. There are people here better qualified than I to comment on that, but I don't see us hitting any limits. Paul.
I wondered if the copper disease was treatable, and if so, how? The disease in the copper transporter.
Well, that's a deficiency of copper transportation, which means you're deficient. A lot of metabolic diseases lead to, well, either a deficiency or a buildup, a deficiency in something you need or a buildup in something that is undesirable. So phenylketonuria is an example of the latter. And both of these classes of metabolic diseases, if they're developmentally sensitive, can be managed to some extent by nutrition. That might be a short-term thing, but if it's a developmental disorder, it has to be managed during the sort of critical phases of development. And that is happening, I think, with a lot of the metabolic rare diseases cases. A longer term view of that is one of the many forms of gene therapy that are currently being trialed, if you understand the genetic basis of what's wrong.
And where do you see the bottleneck in all of this information towards actually the molecular medicine? Where is it going to get stuck, in your view? Is it going to be this accepted, or where's the bottleneck?
So are you thinking of bottleneck in terms of sort of societal acceptability, or sort of more technical?
Well, that's what I'm asking.
Both.
I mean, which is it going to be the problem, or both?
I don't know that there's resistance from society to accept at least being better informed about what might be contributing to particular conditions. I think actually in the UK, I think we've done a fairly good job of educating the public and bringing them along in terms of the positive value of genetic information in medicine, particularly in oncology, and I think more recently in rare diseases. In terms of turning that into sort of new therapies, new cures, new management, the challenge is really integrating this into the NHS from the level of training the next generation of physicians to embrace this, not just in oncology, but across other disease areas. And my wife's a GP, and if I talk to her about this, she'll tell me about the 100 other things that she's up against. She doesn't need genomes on top of that to have to deal with. So there's sort of dealing with things at that level. And then, of course, and there are ongoing projects now at trying to understand how to structure and usefully, in an ethical way, mine information where molecular data such as genome sequences and the many other forms of molecular data are not separated from healthcare data. And they're doing this in FinGen. 10% of their population. We have the Biobank project, which is an excellent project in the UK, and I think aspects of Genomics England and the NHS. We're working on this, but how to bring together multiple sort of complex, multi-dimensional data sets in a way that can be mined. And I'm not an AI expert, so I'm not going to wave my hands in the air and say AI will solve it. But of course, people in advanced data sciences and the big corporates in this area, they're working hard to try and address this with researchers and healthcare organizations. But I think those are the two types of challenges that still remain.
One last question here.
Thank you for that wonderful lecture. I'm just curious what you think is coming down the pipeline with things like liquid biopsy and pharmacogenetics for practice of medicine for people like your wife. Will we be doing just liquid biopsies and sending it off and then having a digital twin for pharmacogenetics modeling before prescribing a drug? Would you see a direction that this was going in?
Yeah, I mean, I can comment on the DNA-based liquid biopsy space, which I've been tracking quite closely. So this has largely been driven by companies trying to develop an early cancer detection method from a blood draw by analyzing the DNA. And as with most technologies, I think it created more excitement than it was ready to justify with hard data. And so that's led to a little bit of skepticism in the field. But as is often the case, I think there is something there in it. I think the first approaches that have gone into large-scale clinical trials are by a long way not the best approaches. There are better approaches now that are more nascent, that will stand to give, I think, better performance, especially at early stage. So I think the liquid biopsy space for cancer detection is a space to watch, actually. I think it's evolving, and it might need another 10 years. Now, there, of course, the vision is to apply it as a screening test for asymptomatic members of the population. Anything applied to screening takes a very long time and a lot of testing to provide, well, to understand if the case is compelling in terms of benefit. So it's too early to call.
Great, and so thank you, Shankar. But before we close today, I just want to say a special thanks to Hilal from Clare Hall, who's really orchestrated this whole event behind the scenes. She's at the back. And thank you to our student volunteers. And most importantly, thank you, Shankar, for being with me.